Now we will discuss about tRNA, the molecular adapter. tRNA is also known as transfer RNA. It is essential link between the language of nucleotides, DNA and RNA, and the language of proteins, amino acids. It is 75 to 90 nucleotides long. These small molecules translate genetic instruction into physical structure by carrying specific amino acids to the ribosome. The clover leaf structure. This tRNA looks like the clover leaf structure. While tRNA folds into L shape in 3D space, its secondary structure is famously described as clover leaf because it looks like clover leaf. This layout ensures that the amino acid is kept at the opposite end of the anticodon, allowing it to interfere perfectly with the ribosome and mRNA. So tRNA folds into L shape in 3D space, and its secondary structure is described as clover leaf. So this is clover leaf. This is clover leaf structure. So tRNA looks like a clover leaf, and it is in 3 dimension. It is formed into L shape, and it is also looks like a clover leaf. So that's why it is also known as clover leaf structure. This is tRNA structure. Here is T loop and D loop, and here is anticodon. Leaf structure. While tRNA folds into an L shape in 3D space, its secondary structure is famously described as a clover leaf because it looks like a clover leaf. The tRNA molecule. The 3' aminoacyl end. This is the business end. This is also known as business end, where the amino acid is attached at the 3' end. Every tRNA ends in the same sequence. Attachment key covalent bond to 3' end. Its mnemonic is CCA, can carry amino acids. The T-arm. The T-arm of tRNA, T-Psi-C. This W-shaped structure is known as Psi or pseudouridine. It is also called as pseudouridine, named for the ribothiamidine, pseudouridine, and cytidine sequence it contains. It is vital for interacting with the machinery. It's the function of T-Psi-C is its ribosome binding key. Ribosome binding key basis. T-Psi-C sequence, its mnemonic is T-arm, tethers to the ribosome. The D-arm. The D-arm of tRNA. It contains dihydrouridine residues. This arm is ID card that the enzyme checks to make sure it has the right tRNA function. Synthetase recognition. Key basis. Its key basis is dihydrouridine. Its key basis is dihydrouridine. D-arm allows detection by synthetase. D-arm allows detection by synthetase. The anticodon loop. It is located at the bottom of the clover leaf. This contains the 3 base sequences that reads the mRNA codon position opposite the 3' end. Its role is matching mRNA codons. Its interaction is base pairing AU and GC. Its activation and energy, the charging. It's the process begun with a specific enzyme called aminoacyl-tRNA synthetase. There is one unique version of this enzyme for each of the 20 amino acids. Energy input. The enzyme uses ATP to activate the amino acid. Specifically, the enzyme must precisely recognize both the correct amino acid and the matching tRNA. Now we will talk about start and stop codons. In mRNA start, there is mRNA start codon and mRNA stop codon. AUG is mRNA start codon. It's also known as inaugurate protein synthesis. It starts protein synthesis happened at AGU start codon. In the eukaryote, it codes for methionine. The mRNA start codon in the eukaryotes, it encodes methionine. In the prokaryotes, the mRNA start codon AUG, it codes for N-formyl methionine, or also known as FMET. This N-formyl methionine, or also known as FMET, stimulates neutrophil chemotaxis. There are 3 mRNA stop codons: UGA, UAA, and UAG. UGA is you go away, you are away, and UAG is also known as you are gone. So these are mRNA stop codon: UGA, UAA, and UAG.